Imagine a world where people live without fear of diseases because they can be instantly deleted as soon as they are diagnosed. Alternatively, imagine a world where the privileged minority rule over subhuman slaves. Today, I'm going to be telling you about a technology that can make either of these situations possible: CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. These are actually DNA sequences that are found naturally in bacteria. CRISPR is used along with the Cas9 protein. The two are next to each other in the bacteria. The Cas9 protein acts as scissors to cut a segment of DNA from the attacking viruses identified by the CRISPR sequences. Once the segments of DNA are stored in the CRISPR sequences, the bacteria can successfully repel attacks by any other viruses of a similar nature. While humans use T cells for immunity against harmful organisms, single-celled bacteria can only use mechanisms that can fit inside one cell. While the original purpose of the system was to defend against viruses, CRISPR can be used to permanently modify genes in other types of organisms as well. CRISPR was first discovered in Japan by Yoshizumi Shino in 1987. Later, Francisco Mojica found these repeating sequences in many species of bacteria. In 2012, Jennifer Doudna and Emmanuel Charpentier began to work with the CRISPR and the Cas9 protein. Soon after this, labs in the Broad Institute, Harvard, UC Berkeley, and Seoul National University managed to edit genes in mammalian cells using CRISPR-Cas9. The CRISPR-Cas9 system has almost unlimited potential, but we have not found a way to implement any of the endless modifications possible yet. However, CRISPR has achieved quite a bit so far. It has been used to inhibit the growth of cancer, store a GIF inside of bacterial DNA, and manipulate the social structure of ants. But, like I said, it can be used for so much more. Treating diabetes patients, eradicating HIV, and eliminating malaria is just around the corner. That being said, should it be used for all of its possible applications? CRISPR is very controversial for a reason. By removing genes that inhibit muscle growth, people can look like bodybuilders without any effort. Countries can create armies full of super soldiers, like those found in fiction stories like Halo. One popular example that is pointed to when discussing the ethics of CRISPR is designer babies. Babies could be programmed to have perfect qualities by changing their eye color, hair color, or height. This is a problem because other qualities that might have advantages might disappear from the human race. Also, the designer babies might be used in a class system, like that of Brave New World by Aldous Huxley, where the most athletic, intelligent, and best-looking people are members of the alpha class, and the epsilon class is dumbed down to the point where they are barely functional and are just used as slaves. Hopefully this will not happen, and it will only be used to stop cancer and other harmful diseases. CRISPR is dangerous in the wrong hands, but if used properly, it is an invaluable resource that will change the world for the better.